What is interphase? Interphase has cells that will divide by meiosis proceed through the growth and synthesis phase of interphase before dividing. This includes replication of chromosomes. At the start of meiosis, a cell contains duplicated chromosomes. Each chromosome is made up of a pair of identical sister chromatids held together at the centromere. Prophase 1. In prophase 1, each pair of homologous chromosomes, one chromosome from each parent, lines up side by side. This alignment of homologous chromosomes is called symphysis. At symphysis, the homologous chromosomes are held together along their lengths. When they are lined up, segments of the chromosomes may be exchanged. This process of exchange of genetic information is an important mechanism for providing genetic diversity and is discussed later in this section. As prophase 1 continues, the centromeres move to the poles of the cell and the spindle apparatus forms. Metaphase 1. In metaphase 1, the pair of homologous chromosomes line up against the equator of the cell. The spindle fibers attach to the centromere of each homologous chromosome. Anaphase 1. In anaphase 1, the homologous chromosomes separate and move to the opposite poles of the cell. Because the sister chromatids are still held together, the centromeres do not split, as they do in mitosis. As a result, a single chromosome, made up of two sister chromatids from each homologous pair, move to each pole of the cell. The chromosome number is reduced from 2 to n. Telophase 1. In telophase 1, the homologous chromosomes begin to uncoil and the spindle fibers disappear. Cytokinesis takes place. A nuclear membrane forms around each group of homologous chromosomes and two cells form. Each of the new cells is now haploid. The phases of meiosis II are similar to the phases of mitosis. The key difference is that the cell undergoes division during meiosis II is haploid instead of diploid. A haploid number of chromosomes line up at the equator during metaphase II. During anaphase II, the sister chromatids are pulled apart at the centromere by the spindle fibers. The chromosomes move toward the opposite poles of the cell. The chromosomes reach the pole during telophase II, and the nuclear membrane and nuclear reform. At the end of meiosis II, cytokinesis occurs, resulting in 4 haploid cells, each with n number of chromosomes. Prophase II. Prophase II begins immediately after cytokinesis, the splitting of the diploid gamete into haploid daughter cells. In order to prepare for the next division, prophase II condenses chromatin first into chromatids, and then tightly moving chromosomes. Simultaneously, the nuclear membrane dissolves, leaving an open area of cytoplasm in which a network of proteins have enough space to create pathways reaching from one side of the cell to the other. The spindle apparatuses pinch in. Metaphase II. Chromosomes line up separately on the equator of the spindle, kenotorches pointing towards opposite poles. Anaphase II. The chromatids of the chromosomes finally separate, becoming chromosomes in their own right, and are pulled to the opposite poles. Or, the pulling apart of either homologous chromosomes or sister chromatids is called distraction, and occurs during anaphase of meiosis I or meiosis II. Telophase II. Daughter chromosomes reach the opposite poles. The two cells invigate and form 4 daughter haploid cells. They uncoil and form a chromatin network. Nuclear envelope and nucleus form around the chromatin network again.